Hello, my name is Abhay Hastir, and my topic is the effect of different sugars on carbon dioxide production by fermentation of yeast. Introducing yeast. Now, we come to a question: what is yeast? Yeast is an unicellular organism which is used in many processes such as curd making, alcohol production, and many more. It produces alcohol and carbon dioxide by consuming sugars, and this process is called fermentation. They can either perform aerobic respiration or anaerobic respiration depending on the oxygen rate. There are many types of yeast, but we will use yeast commonly known as baker's yeast, or also known as Saccharomyces cerevisiae. I believe that when the graph is made, there would be an exponential growth in certain type of yeast, mainly glucose type. Here is the image of fermentation. What is the purpose of our study? We aim to show that different amounts of sugar can be changed by processes fermentation, and it can affect the carbon dioxide production. There comes another question, that is, how it would change the rate of fermentation and what would happen to the yeast by changing the sugar solution. The material used were 14 test tubes and their respective racks, a prepared bath for heating system, and the solution used are glucose, sucrose, maltose, galactose, fructose solution, and water. Now the method section. Test tubes and hot tub bath were prepared. 7 milliliter of sugar solution was added to each test tube. 8 milliliter of yeast solution was added to its test tube by reverse pouring method respectively in a different test tube. The test tubes were then introduced in the heating system, that is the hot tub bath, in a test tube rack respectively. The amount of growth was noted after every 5 minutes interval. The independent and dependent variables were the amount of yeast solution and the amount of carbon dioxide produced respectively, and the control was water. Next, experiment was repeated many times, and the results show exponential growth. It is found that exponential growth occurred only in certain types of yeast solutions such as sucrose and glucose. The results show that there was 2 milliliter growth after 5 minutes interval. We found that disaccharides such as galactose took longer time as compared to monosaccharides such as glucose. Now that we know that formation of bonds play an important role, as glucose is simpler sugar while galactose is complex sugar which is converted to glucose in the process fermentation. Overall, the sugar solutions such as maltose and galactose in the first part did not reach above 5 milliliter, while the others showed exponential growth, as shown in the graph. They did not exceed 14 milliliter. Now there are some limitations to our experiment, such as difference between two test tubes after 45-minute controls cannot be noted due to test tube being fully not visible. The time to enter the test tube contains solution may have already begun fermentation and can alter its result. The repetition method did not always work as the data did not match every time the experiment was performed. Thank you. This is the reference page.